Okay, so the diagrammatic representation of anatomy of a muscle fiber showing a sarcomere. Okay. Now the structure of contractile proteins, myosin and actin. Now actin, 2F filaments and monomeric is globular, and tropomyosin also double, 2 filaments. Now a complex protein troponin is distributed at regular intervals on the tropomyosin. Now in the resting state, a subunit of troponin masks the active binding sites for the myosin on the actin filaments. Okay. Now meromyosin, monomer unit of myosin. There is LMM, light meromyosin, and heavy meromyosin. So the heavy meromyosin has head and short arm, and light meromyosin is the tail part. Okay, not the tail part. Each meromyosin has two different parts: a globular head with a short arm and a tail. Okay, yeah. The tail is the light meromyosin, right? And head is the heavy meromyosin, right? Now you see, the globular head is an active ATPase enzyme and has binding sites for ATP and active sites for actin. You see the cross arm. Mechanism of muscle contraction. So mechanism of muscle contraction is best explained by the sliding filament theory, which states that the contraction of a muscle fiber takes place by the sliding of the thin filaments, that is actin over the thick filaments. Then muscle contraction is initiated by a signal sent by the nervous system CNS via motor neuron. Now a motor neuron along with the muscle fibers connected to it constitute a motor unit. Now okay, so motor neuron and muscle fiber is motor unit. Now motor unit and sarcolemma of the muscle fiber is neuromuscular junction or motor end plate. Okay. Now a signal reaching this junction releases neurotransmitter acetylcholine, which generates an action potential in sarcolemma. Now this spreads through the muscle fiber and causes the release of calcium ions into the sarcoplasm. Now increase in calcium ion leads to the binding of calcium. Now increase in calcium level leads to the binding of calcium with a subunit of troponin on actin filaments, and there remove the masking of active sites for myosin. So utilizing the now. So this is it. Now ATP hydrolysis, that is formation of cross bridge, that is phosphate, inorganic phosphate and ADP. So this is formation of cross bridge. Now this pulls the attached actin filaments towards the center of A band. Now the Z line attached to these actins are also pulled inwards, thereby causing a shortening of the sarcomere, that is contraction. Okay, so during shortening of the muscle, that is contraction, the I bands get reduced, A band retains the length. Okay, now the myosin releasing ADP and inorganic phosphate goes back to its relaxed state. Now a new ATP binds and a cross bridge is broken. So when ATP hydrolysis, that is cross bridge is formed, formation of cross bridge, and when ATP binds, then cross bridge breaks. The ATP is again hydrolyzed by the myosin head, and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filaments. Okay, resulting in the masking of the active filaments. So this causes the return of Z lines back to their original position, that is relaxation. Okay, repeated activation of the muscles can lead to the fatigue. Now muscle contains pigment, red color pigment, that is myoglobin. So there are red muscle fibers, white muscle fibers. Red muscle fibers, these are aerobic aerobic muscles and anaerobic muscles, these are white muscle fibers. So skeletal system. Now skeletal system consists of a framework of bones and a few cartilages. Bone and cartilage are the specialized connective tissues. Okay, so bone very hard, matrix due to calcium salts, and cartilage slightly pliable matrix due to chondrine salts.